Hello and welcome to Bio40 Pre-Lecture 27. Let's continue talking about gene regulation in bacteria. Last time we discussed the lac repressor system. When lactose isn't around, the lac-I gene produces a repressor that binds the lac operator, preventing RNA polymerase from binding the promoter and transcription from occurring. But when the inducer allolactose is present, it stops the repressor from binding the operator, so transcription of the structural genes can occur. This is called an inducible system. But when a mutation inactivates the repressor, or the operator is modified such that the repressor cannot bind it, the structural genes are always expressed. This is constitutive expression. Now let's take a closer look at the lac repressor protein. On the right is a cartoon structure of it. The 4 different colors represent 4 polypeptides. The repressor functions as a tetramer. Specifically, it's a homotetramer. Each subunit is the same polypeptide, the one coded for by the lac-I gene. Now we know the repressor must bind the lac operator in the absence of allolactose, and it must bind allolactose, and this interaction should cause a change that stops it from binding the DNA. So that's 3 things the repressor needs to do. If we were collecting lac-I mutants, we should be able to find some that only affect one of these functions. What would each of those mutants look like? Well, if the protein can't form tetramers, and monomers do nothing on their own, this is equivalent to completely inactivating the gene. You'll get the same constitutive lac expression as with the lac-I minus mutants we talked about last time. But mutations that affect DNA or allolactose binding will be a little different. Let's look at mutations that prevent the repressor from binding the inducer. It still forms tetramers, and it still binds the operator, so in the absence of allolactose, transcription is blocked like it should be. But if allolactose is added, the repressor won't bind it. It will still sit on the operator and block transcription. Expression is now uninducible. It will not occur under any condition. We call this mutant a lac-I superrepressor, and we use a superscript S in our notation. Now let's see what happens when we try to complement the lac-I superrepressor with wild-type lac-I. In the absence of inducer, the superrepressor has a wild-type phenotype anyway, so we don't expect any difference then. But what about when lactose is present? Well, all the wild-type repressor will bind allolactose and no longer interact with the operator. But the superrepressor will ignore the inducer and continue to bind its operator, as well as the one on the other DNA molecule. Therefore, we say the superrepressor can act in trans. With it binding both operators, the structural genes won't be transcribed from either DNA molecule, so expression is uninducible. Since we have the superrepressor phenotype in this heterozygote, we can conclude that it is dominant to wild-type. So we've covered wild-type lac-I, we've covered lac-I minus mutants, which lead to constitutive expression of the lac operon and can be the result of absent or misfolded protein or problems in forming tetramers. And we just saw the superrepressor, which still binds the operator, but not the inducer, preventing synthesis under all conditions. But then we have mutants that can still form tetramers, but can't bind DNA. If it can't bind the operator, it can't block transcription in the absence of inducer, so expression is constitutive. But how is that any different from the lac-I minus mutants we first talked about? Think about this, and we'll discuss it in class. Okay, enough about lac-I. This isn't even the only system that regulates the lac operon. Many operons involved in catabolism, the breakdown of complex molecules to produce energy, are regulated by glucose. Glucose is the preferred energy source for E. coli, along with many other prokaryotes and all eukaryotes. So if the cell is in an environment where both glucose and lactose are around, it will metabolize the glucose and prevent expression of the lac operon. But this repression is indirect. It's mediated by the molecule CAMP, or cyclic AMP. Cyclic AMP is produced from ATP, but only in the absence of glucose. So high glucose levels mean low cyclic AMP, and low glucose means high cyclic AMP. Cyclic AMP binds a catabolite activator protein, or CAP. As the name implies, CAP is an activator. It helps RNA polymerase bind the promoter. But it's only active when cyclic AMP is present. So when CAP interacts with cyclic AMP, the activator binds the site on the DNA close to the LAC promoter. Then CAP helps RNA polymerase bind the promoter, greatly increasing transcription of the LAC operon. So optimal expression of the LAC operon requires both the presence of lactose and absence of glucose. Let's look at the magnitude of the effect. We'll use no lactose plus glucose as our baseline 1x. Remember, expression should never be 0 in this case, because the cell needs at least a little LAC permease and beta-galactosidase to sense the presence of lactose in the first place. Now if we add inducer, expression goes up 20-fold. And that's using the strong synthetic inducer IPTG that we tend to use in lab environments. That's a decent change, but it's nothing compared to the 1,000-fold increase you get with both the inducer and no glucose. So that covers our discussion of the LAC operon. But catabolic genes aren't the only ones that use operons. Genes involved in anabolism, the synthesis of biological molecules, are also found in operons. We've seen before that many genes are typically required in the synthesis of molecules like vitamins, nitrogenous bases, and amino acids. If a given molecule is needed, every gene involved in the process needs to be transcribed. So it makes sense to group them in an operon, where they can all be regulated as a single unit. For our model system, we'll look at the TRP operon. The TRP operon includes 5 genes, TRPA through TRPE, that are necessary for the synthesis of the amino acid tryptophan. In front of those genes are the promoter and operator, like you've seen before. But there's also this leader region, which is new. We'll come back to it in a moment. So when does the cell want to express tryptophan, and when doesn't it? Well, tryptophan is an amino acid, and the cell is making proteins all the time, so there's likely to be a constant demand for it. The only time the cell wouldn't want to make tryptophan is if it already had plenty of it available. So the presence of tryptophan should downregulate the expression of these genes. This is indeed the case, and it happens in 2 ways. One way involves a repressor, much like we've seen before. The other is a completely new process called attenuation. The TRP repressor works just like the basic repressor-co-repressor system we talked about before. The protein is encoded by the gene TRPR. This gene is located far from the TRP operon, but since proteins act in trans, it doesn't need to be close by. In the absence of tryptophan, the repressor doesn't do anything. Transcription occurs, and the cell makes the amino acid it requires. But when plenty of tryptophan is available, it binds the repressor, and the repressor can then bind the operator. This stops RNA polymerase from transcribing the structural genes, and the cell doesn't waste energy synthesizing an amino acid it already has. But a funny thing happens when you look at TRP operon expression in TRPR mutants. In a wild-type cell, the presence of tryptophan reduces the expression of the TRP operon by 92%. However, if you knock out the TRPR gene, the presence of tryptophan still reduces TRP operon expression by 2/3. So some other system must be downregulating expression in response to tryptophan. This system is called attenuation. Rather than blocking the polymerase from ever starting transcription, attenuation involves early termination of transcription. Recall that we talked about 2 forms of transcriptional termination in E. coli, row-dependent and row-independent. Attenuation makes use of row-independent termination, wherein this hairpin, or stem loop structure, causes RNA polymerase to stop transcribing. The first evidence for attenuation came when researchers saw that there are 2 alternate RNA transcripts produced by the TRP operon. One is quite short, indicating that transcription was halted not long after it began, but the other is much larger. It includes all 5 structural genes. The less tryptophan there was available, the more the longer transcript was favored. This is all due to this leader sequence, a small region that comes after the promoter and before the structural genes. Let's look closer at it. The leader region contains a very short gene referred to as TRPL. The polypeptide it codes for has just 14 amino acids and serves no function, but it contains adjacent tryptophan codons. So if tryptophan levels are low, the ribosome will stall there, waiting for tryptophan tRNAs to arrive. But if there's plenty of tryptophan around, the ribosome won't have any trouble finding tryptophan tRNAs, and it will speed right past these codons. The RNA will adapt alternative secondary structures based on whether or not the ribosome pauses, and these structures determine whether or not transcription is terminated early. It all comes down to these 4 regions of the transcript. Region 1 is a coding region rich with tryptophan codons, where the ribosome will pause if tryptophan levels are low. Region 3 is capable of forming a hairpin with either region 2 or 4, and transcriptional termination is determined by which hairpin forms. If there is plenty of tryptophan present, the ribosome will not pause at region 1, and will move over region 2. With 2 inaccessible, regions 3 and 4 will pair, forming a termination loop. This will cause RNA polymerase to fall off the DNA, ending transcription before the structural genes are transcribed. However, when there is little tryptophan around, the ribosome stalls at the tryptophan codons, and doesn't reach region 2. As a result, regions 2 and 3 pair, forming an anti-termination loop. This loop does not stop transcription, and it leaves region 3 unavailable to form the termination loop with region 4. The polymerase is allowed to continue, and the structural genes are transcribed. So those are the 2 levels of regulation for the trip operon: a repressor-co-repressor system, and attenuation. Think about what sorts of mutations you could get that would affect attenuation. Would they act in cis or in trans? Also, do you expect to see attenuation in eukaryotes as well, or just in prokaryotes? We'll discuss this all in class. Before we wrap up, I quickly want to run through some other methods of regulation you'll learn more about as you continue studying biology. One type that I find really cool are riboswitches. Riboswitches are pieces of RNA that can directly interact with effectors, such as inducers or inhibitors, and these interactions can affect the formation of other structures. The part of the riboswitch that interacts with the effector is called an aptamer. When the aptamer binds the effector, the structural change could lead to the formation of a termination loop that halts transcription, or it could leave the ribosome binding site inaccessible, blocking translation. These are both forms of negative regulation, but aptamer binding could just as easily prevent termination loops from forming or make ribosome binding sites accessible. It all depends on the specific situation. Additionally, there are small RNAs, or sRNAs. sRNAs can base pair with ribosome binding sites, blocking access to the ribosome and preventing translation, though these too are capable of positive regulation. Sometimes, the default is for the transcript to block its own ribosome binding site with a stem loop, but the sRNA can disrupt this structure, allowing the ribosome to bind and translate. Another way sRNAs can function is to lead to the degradation of the mRNAs they pair with before translation has a chance to occur. Finally, there is antisense RNA. The normal mRNA is known as sense RNA, so antisense RNA is made when the other DNA strand is used as a template. The sense and antisense RNA strands can pair together, and this pairing can block translation. Also, double-stranded RNA is typically recognized by the cell as foreign and quickly degraded, and the actual process of making antisense RNA can even interrupt the transcription of the sense RNA, since transcription should not be occurring in both directions at the same time. That's it for bacterial regulation. Next time, we take a look at how gene regulation works in eukaryotes.